All right, everyone. Welcome to the review session for this week. I know it was a snow day. I hope you guys had a good snow day. But we're going to be talking a little bit about proteins and enzymes and how proteins work in the context of what their actual job or what their actual role is. And the key thing to think about here is the protein structure and how that relates to the protein function. Um, we're going to record this like us going through the slides, and then we'll stop the recording and we'll like open it up for like more like open Q&A if you guys have questions about the problem set or anything like that. But also feel free if you're confused about anything on the slides, how that pertains to anything else. If you have any kind of questions, feel free to raise your hands, unmute, anything like that. Type in the chat, Rosella, if you can watch the chat while I'm going, and then we can swap. But
Yes.
uh, yeah, feel free to ask those questions. Stop us at any time. We'd be happy to explain or clarify. All right, so let's get started. I'm Aman, and this is Rosella. And yeah, let's go. All right, so the first thing we want to talk about is protein ligand interactions, right? So you guys probably all have seen this image. This was from your what's it called? Your problem sets, as well as the lecture. Dr. Francis had this on the slides. And what we're showing you here in this picture is like an interaction between a protein, which is this gray uh, line, and a ligand, which is this red thing right here. It's cyclic AMP, right? So a ligand is something that a protein binds to, regardless of like what that protein's function is or what it's going to do to the ligand. If it binds to that ligand, that is a ligand for the protein. Here in this diagram, it's like shown that like the ligand is binding in this nice little pocket, like the physical structure is like kind of this like cup-shaped pocket. That's not always true. This just happens to show a binding pocket, but it doesn't have to be that pocket shape. It can be any kind of surface on the protein that binds to whatever ligand. However, the most important thing here is the bonds between the ligand and the protein, right? To the binding surface on the protein. So the ligand, in this case, the cyclic AMP, has to be correctly oriented, and the protein has to be correctly oriented, such that all these bonds that we see here, the electrostatic attractions, the hydrogen bonds, everything else, those bonds need to occur. Otherwise, the ligand and the protein are not going to stick together, right? And the key thing here is like the R group interactions. If you remember, like your tertiary and quaternary structure, the again the R group interactions are the key interactions that define tertiary and quaternary structure, and then moving forward, like define like how that tertiary quaternary structure interacts with other proteins, ligands, whatever the ligand is. Um, so remember, these are like all kinds of bonds. We're talking about like polar bonds, first and foremost, as like the hydrogen bonds, ionic bonds. You have the electrostatic interactions. You also have like the van der Waals interactions, which are the non-polar to non-polar interactions. And then in some rare cases between protein ligands, you might have covalent bonds in the form of disulfide bridges. However, since that's a really permanent, strong bond, it's not really likely that, or it's not common to have a ligand and a protein bind together so permanently, right? So again, key things to take away here is that the chemistry and of the bonding needs to be correct, and that comes both in the chemistry, comes via the chemistry of the amino acids involved in that binding process, as well as their physical structure, right? So if I swap this serine with something like a phenylalanine, which is a really large, bulky, non-polar structure, you have both a loss of the polar chemistry of the serine, which means that these hydrogen bonds are no longer going to occur, and you have now a really large, bulky amino acid there, which might stick out in the binding pocket and make it so cyclic AMP can't physically fit. That's something called steric hindrance, right? Hindrance due to space constraint. Does that make sense to everyone? Are there any questions? Anyone wants to put anything in the chat or raise their hands? Okay. Also, if anyone wants me to like slow down or anything, feel free to let me know. But yeah, so again, the ligand, the protein, they have to be oriented in the correct way such that the corresponding interactions uh, based on size and chemistry can occur. All right. The next thing I want to talk about is affinity, right? So this is affinity between the ligand and the protein. Affinity is just it's a term for the strength of the interaction. So the greater the affinity of the protein for the ligand, the stronger that protein is going to like latch onto and hold on to that ligand. And there are two big things that kind of determine that affinity. One is the number of bonds between the protein and the ligand, and the second one is the strength of those bonds. So, like I said here, um, what is it? Disulfide bridges. The covalent bonds are highly uncommon because they're so strong, right? And sometimes that can be too much affinity for a protein and a ligand. Yeah, that can be too much affinity between a protein and ligand. In some cases, that's necessary. So you might see those really strong disulfide bonds. But if you count, like, for example, like 10 hydrogen bonds between one protein and ligand versus like 3 or 4 van der Waals interactions and maybe like one ionic bond between another protein and a ligand, you know, the one with the more bonds and the stronger bonds, the hydrogen bonds, will have a much stronger, like a much greater affinity. Does that make sense to everyone? I hope. Any questions? All right. Um, so the strength of those bonds, especially, is what comes down to the type of the bond and the angle of the bond. So that's again what we talked about here. Here, with the size and chemistry, if you have bonds that are really strained angles, they're not going to be able to. They're not going to be as strong, depending on the size of the amino acids involved, the size of the ligand, and how that interaction takes place. If you have something that's like really large and bulky and doesn't fit super well, and the bond that you're forming is at a very sharp angle, that bond is much more liable to break, and therefore the affinity for the protein to the ligand is much lower. And then, like I just mentioned, the type of bond, covalent, non-covalent, and you know all the subtypes of non-covalent bonds. Those two things are really big at play here. All right, any questions? All right. Perfect. And now we're going to move on to a little bit of math, which is most of the time not anyone's favorite, but it's not too bad. This is KD, or the dissociation constant, right? So KD is a way to actually represent affinity and describe protein ligand interactions. It's like a numerical, quantifiable way to represent affinity. The KD, or dissociation constant, is the amount of free protein that you have times the amount of free ligand divided by the amount of ligand protein complex. So the ligand and protein stuck together. So if you have a ligand that has, or a protein that has a high affinity for a ligand, then you're going to get a lot of protein stuck to a lot of ligand, which means that the amount of complex that you have is going to be really high. So if you're increasing the amount of complex, you're increasing the denominator in the KD equation, which means that your KD is going to go down. So again, high affinity means high amount of the complex, which means that you have a low KD value. So yeah, so again, like a smaller KD here, this corresponds to a tighter protein and ligand binding interaction, or greater affinity. Keep in mind that it is an inverse relationship. There's also something that you might see especially if you're like using Google or anything like that, called Ka, which is Ka, right? So yeah, association constant, which is the inverse basically, and that doesn't is like a direct relationship to affinity. So if Ka goes up, affinity goes up. However, in biochemistry and biology, in the context of this class, we're going to talk about KD. It's a lot more relevant to like the scientific nomenclature and the context that we're using. So again, KD goes down, affinity goes up. Any questions about KD?
So they're always opposite.
Yes, KD and affinity are opposites, and it's always like a KD is a given value between one protein and one ligand, right? So the KD is like the KD for protein A to ligand A, right? No matter how much protein you have, no matter how much ligand you have, the KD is a constant value that represents the affinity.
So I have a question. So what happened, I don't know if this is, in fact, this is gonna happen, but I'm just saying in scenario where it's KD and definitely they are equals, or what happened?
Sorry, say that again.
Is there a scenario where KD like is exactly equal to affinity?
Well, so
Affinity.
affinity is not like, so in this scenario, KD is like the actual number, right? KD, so affinity is, you're not like, you don't have a number for KD is the number that represents affinity, right? So it's not so much that you're comparing the KD of a protein and ligand to the affinity between the protein and ligand. You're comparing the KD between two sets of proteins and ligands, right? So for example, if the KD for protein A to ligand A is 0.5, but then the KD for protein B to ligand B is 2, right? The one with the lower KD, which is the 0.5, which is protein A and ligand A, has a greater affinity for each other. So protein A and ligand A will stick together much better than protein B and ligand B will stick together. Does that make sense?
Oh yeah, thanks.
Lovely. Perfect. All right. Any other questions? That was a great question. Appreciate that. All right. So this is Rosella's, this is going to like lead into what Rosella's going to be talking about with enzymes, but this is like a little bit of context to help to like actually visualize and understand KD. This is kind of like what we were just talking about. If you want an enzyme to be binding strongly to a substrate, right, which would be the ligand, this is again like, let's say you have enzyme A and ligand A or substrate A. A high affinity would mean a low KD, and you really want the enzyme, if you want the enzyme to do its work really well, you want the enzyme to pick up substrate really well. You want it to like grab all the substrate it can and quickly turn that into product, right? So the KD between the enzyme and the substrate is really low because you want the enzyme to quickly pick up the substrate and securely hold on to it. But as soon as that substrate is turned into product through whatever catalytic activity, you want it to spit out the product, right? You don't want it to stay holding onto that product. So the KD for the between the enzyme and the product should be much higher because you want the enzyme to spit out that product and kick it out. Right? Does that make sense? So that's like another scenario where instead of comparing enzyme A and ligand A to enzyme B and ligand B, now we're talking about enzyme A and substrate versus enzyme B and product. You want the enzyme A and substrate KD to be really low, and the enzyme A and product KD to be really high. Does that make sense, everyone? I'm going to pause for questions. All right. Lovely. Yeah, this is kind of just like showing what I was just talking about, but I don't know if this leads into Rosella, what you want to go through, but in this scenario, can everyone see my mouse?
Yes.
So in this scenario, you want the enzyme to pick up the substrate really quickly and hold on to that substrate strongly. So here, between this interaction to, or this state to this state, you'd want a really low KD to run this. And then you have catalysis occurring, and now you have a different product, right? The product is no longer the same thing as substrate. And then you want the enzyme to spit out the product as fast as possible. So you want a really high KD to govern this this process, the spitting out process. All right. Now we'll go into enzymes. Rosella's going to take over.
On can I ask a quick? Could you go back again over there?
Yeah.
I think on one of the problem sets they did ask a little bit about the binding interaction between enzyme and substrate.
Yeah.
When the interaction between the enzyme and substrate is stronger than the binding interaction between enzyme and product. So could you explain a little bit more about that question?
I don't know what question that is off the top of my head. I would be happy to go over that. I'm just gonna like push that to the end to go over the specifics of the question. But essentially, like that gets from what I remember, that gets at like the heart of what we're talking about here, where you want the enzyme to hold on to the substrate right? Really strongly. And then once that substrate is turned into something else, which is the product, right? Remember that the product and the substrate are not the same thing. So they're chemically different, and therefore they'll have different like strength of interaction. They'll have different chemistry of interaction and therefore different strength of interaction with the enzyme. So you want the enzyme to hold on to the substrate really strongly until that substrate is turned into enzyme, and give it time for that substrate to turn into enzyme. And as soon as it turns into, or substrate to turn into product, and as soon as that substrate turns into product, you want it to let go, right? So that would be you want a high affinity between the enzyme and the substrate and a low affinity between the enzyme and the product.
Could you repeat one more time with that sentence?
Yeah, you'd want a high affinity between the enzyme and the substrate, and then a low affinity between the enzyme and the product. Does that make sense?
Yeah, thanks.
Alrighty.
Okay, enzymes. Go on to the next slide. So what are enzymes? So enzymes are catalysts and they carry out chemical reactions. So catalysts basically describe something that like makes something go faster. And enzymes, what they do is they make chemical reactions go faster, basically. Like from products to reactants, they make that process go a lot faster. So without enzymes, a ton of chemical reactions that are essential to us would not happen. For example, there are a ton of enzymes involved in what just happened. Oh, sorry. My screen just went white. Okay, I'm okay. There are a ton of enzymes involved in making ATP, so making energy. So enzymes are basically everywhere. They're very important. They make and break covalent bonds, right? So for example, a covalent bond that we talked a lot about in class are peptide bonds. And the making of those peptide bonds, which is the links in between the amino acids, that is made through an enzyme. And that enzyme is the ribosome. So the ribosome, like we talked about, it's made up of proteins and RNA. rRNA, ribosomal RNA, and the thing that is actually that actually has the catalytic component in the ribosome is the RNA. So not all enzymes are made up of only 100% protein, because as we know, ribosomes are an example of a ribozyme, which is an enzyme that uses RNA as a catalytic component. Yeah, another example of when enzymes are important are during RNA processing, when introns are removed from mRNA so that you only have the coding parts of the RNA that get sent to the ribosome. Any questions on that? Okay, move on. Okay, so enzymes, the way they work is that they bind to the substrate, and then basically there are different ways that enzymes orient the substrate that we can talk about later, but basically bind to the substrate. The enzymes undergo conformational changes during the reaction, and then it makes the products form. And then after, when it lets go of the products, the enzyme returns back to the state it was before. So the enzyme before the reaction and after the reaction stays the exact same, because it's not involved. It helps the reaction go through, but it's not involved in the reaction. And the enzyme does go through chemical conformational shifts, but in the end, it remains the same as before. Also important because the enzyme needs to be able to catalyze multiple chemical reactions. So it's not just like it catalyzes one and then it can't work anymore. It needs to be reusable. Also, just like Aman was saying before, it is important that enzymes have higher affinity for reactants than the products. So when we were talking about, so KD and affinity. So high affinity means that the enzyme and the substrate are more likely to bind, right? So they have high affinity. If something has low affinity, that means that they are less likely to bind, right? And so when we talk about KD, we're talking about the constant of dissociation constant. And dissociation means to to separate, right? So that's why when you have higher affinity, it means that the enzyme and the substrate or whatever proteins, when you have higher affinity, that means that they're less likely to dissociate, which is why the KD is lower, right? So when you have two proteins that have low affinity, which means that they aren't as likely to bind together, right? The dissociation is higher because they don't have a strong chemical bond, so they're more likely to dissociate. So that's the whole inverse relationship, just to reiterate, because I know a lot of people get confused about that. And with enzymes, it's important that the substrate has higher affinity, which means lower KD, than the relationship between the enzyme and the product, right? So the enzyme and the product have a lower affinity, so they have a higher KD, right? More likely to dissociate. This is important because the enzyme needs to be able to bind the substrate, like needs to find the substrate, bind, and then needs to hold on to the substrate until it turns into the product. And when you have the product, you want the enzyme and the product to let go of each other, because the product needs to go do its function. If the enzyme and the product are bound together for too long, then that's going to make the reaction just go slower, and the product can't go fulfill its function. Yes, any questions on that? Okay. Why are enzymes useful? We sort of already went over this before, but enzymes, they speed up reactions, right? That would take way too long if they occurred spontaneously. And they increase the frequency of chemical reactions, meaning that you get more products, and yeah. Something very important about enzymes is that they are able to be regulated, and they can be regulated through multiple ways. One is that they can be regulated through transcription. So if your cell really needs an enzyme, it can go transcribe it and translate it. And then if it doesn't need an enzyme, it just won't transcribe and translate it. And then if you have too much of the enzyme and you don't need it anymore, the enzymes can also be tagged to be destroyed through the proteasome. Also, if enzymes are needed in a different area in the cell, they can be moved around the cell. So it's really important to be able to have regulation in chemical processes, just to make sure that the cell is doing everything right. More regulation is usually better, less of a chance that something will go wrong. And so, yeah, so enzymes allow for this specific regulation of when chemical reactions occur and where.
I have a quick question about that.
Yeah.
Since you're talking about a chemical reaction, so what happens when we have a mutation that decrease or maybe increase the KD of the enzyme, like in the product associated?
Okay, yeah.
How does it affect?
Yeah, so if the KD increases, right? The KD increases, or let's say that the KD between the enzyme and the product decreases. All right, so if the KD decreases, we know that the affinity is increasing, right? So if that happens, that means the enzyme and the product are going to be together bonded for longer, right? Because they have more affinity, less likely that they dissociate, so lower KD. And that means that the reaction is going to go slower, right? Because if the enzyme and the product are bound together for a longer amount of time, that means that it's going to take more time for then the product to be released and go do its function. Did that sort of answer your question? I can do another sort of example.
No, that was perfect. I think
Okay.
you did really good. Thank you so much.
Okay. Also, another thing to note is that more affinity doesn't always mean doesn't always mean a good thing. So because I think some people sometimes get the wrong idea that more affinity means good, but it doesn't, because you need to have these transient protein interactions in the cell, right? So if the product had more affinity with the enzyme, that's bad, because you don't want them to have more affinity, right? So less affinity is actually better in that situation. So don't confuse yourself when you think about affinity, that it's not bad or good. It's like that whole, like Laura was saying, like the affinity needs to be just right for that specific interaction. Yeah.
Yeah, that makes sense. Thanks.
Okay, you can move on to the next slide. Okay, so this is just one of those reaction process diagrams. I'm sure you've seen a ton of these. On the y-axis, you have the free energy. So it's talking about the chemical potential energy of the bonds in between the molecules of the reactants and the products. And then on the x-axis, you have like the progress of the reaction from reactants to products. So between to get from the reactants to the products, you need to go over a bump that's called the activation energy, right? So this is a place where the free energy increases a lot, goes up to the activation energy, and at the top of the hill of the activation energy is something called the transition state. And this transition state is an unstable state, and what enzymes do is stabilize the transition state and decrease the activation energy. Okay, so in the solid line, that's the reaction process for an uncatalyzed, so no enzyme reaction. And then in the dotted line, that's the reaction process for a catalyzed reaction with enzymes. So you can see that the activation energy is much lower with the enzyme, and the height of this activation energy is directly correlated with how fast the reaction is going to go. So we can see that the activation energy without the enzyme is much higher than with. So that means that the reaction is going to happen much faster with the enzyme, which is, we know that that's true, but yeah, so because the activation energy decreases, that's gonna let the reaction go by faster. Over here, you can see that this is an exergonic reaction. So if you guys remember, exergonic means that you have like a net release of energy, and then endergonic, which is where the reactants are lower than the products, that's going to be a net input of energy. So even though this, because the reactants have more energy than the products, that means that this is going to be like most likely a spontaneous reaction. But even spontaneous reactions sometimes need an enzyme in order to get to the products, because it could take a very long while for that spontaneous reaction to actually occur. Yeah. Any questions?
Nope.
Okay. Reaction process. Okay. So this, I think this is sort of reiterating what I said. There is always a transition state, which is the top of that activation energy hill. And that is a place where the molecules are unstable. There's a lot of they're at a high energy state. So what the enzyme does is lowers and stabilizes that transition state, also lowering the activation energy, makes the reaction go faster. And yeah, below is just reiterating the whole like spontaneous, non-spontaneous, endergonic, exergonic sort of thing. Again, enzymes are really beneficial because they lower the activation energy, which means that the reaction goes by faster and it is more energy efficient. So there is less energy needed in order for the reaction to take place. So you're wasting less energy. And there are different ways in which the enzymes bind the substrate and work. You don't need to go too much into detail on these. This is just like a very brief overview. One way is that the enzyme binds the substrates in a way that orients them to just make them undergo the reaction from products, reactants to products. So the first one is just like spatially orienting them in order to cause the reaction to happen. The second one is basically the enzyme is binding the substrate and there is a rearrangement of electrons, which creates partial positive, partial negative charges, and it just causes the reaction to take place faster. And then the last one is that the enzyme sort of strains the substrate and forces it into its transition state. Which is that transition to that, which means it makes it go to the transition state faster than it would without the enzyme. It sort of forces it to go there. And then that ends up causing the reaction. So yeah, you don't really need to know you don't need to really know too much detail about this, just basically what's on the slide is basically the extent of what you sort of need to know these three things. But you can do more research into these later. But I think you'll talk more about these in biochem classes. Yeah.
Yeah, basically, I think it's just like know that there's like a multitude of different ways in which enzymes work, and what's it called? And enzyme activity is like highly regulated, right? So regardless of like, depending on the way that the enzyme carries out its activity and things like that, there are like things like other cellular processes that step in to turn enzymes on and off, and that's leading into what I'm going to talk about in a second. Also, I just saw the question in the chat. Are there any ways that enzymes can slow down the reaction? Not real, like the function of an enzyme is to decrease like the activation energy, right, which like reduces the energy like demand in order to like kind of kickstart the process. It's because you kind of have to pour energy in to get your reactants to that transient high energy transition state, right? So you're by reducing the amount of energy it takes to do that, by creating a favorable state, like by favoring the transition state and stabilizing transition state, the enzymes will actually speed it up by reducing the energy threshold. There are ways in which you can slow down a reaction, but enzymes aren't going to be the things to do that. The number one thing is just like a barrier. So for example, to slow down the rate of gene expression, you have like the nucleus, which separates ribosomes and things like that from like the mRNA and the DNA and things like that. So there's like a lot of like physical barriers. First and foremost, there are ways to sequester certain enzymes or products, reactants, whatever, in such ways that you can slow down a reaction. But enzymes are not really the way you'd be slowing things down, right? You can also reduce the temperature, for example, and that will reduce the amount of energy that you have freely available to put into the system to bring the reactants to the transition state. But again, that's not like quite enzymes. Does that answer your question, Jen? Yeah. All right. So yes, like I just said, enzyme activity is highly regulated, and that's what I'm going to be talking about now. And one of the most important things is the idea of allostery, right? Allostery is a regulatory process through which an enzyme will have essentially multiple binding sites for ligands. However, if the enzyme has some specific like role or function, not every binding site has to be about that function or directly bring about or carry out that function, right? So enzymes can also have like secondary sites, essentially binding sites, which are regulatory sites where you can have allosteric binding. Allosteric binding is basically when a ligand binds to an enzyme and carries out some sort of conformational change. It's really important that you understand that it's a conformational change in the enzyme because, again, structure equals function. So if you're changing the structure of the enzyme, you're changing the function of the enzyme. So the binding of a ligand to the allosteric site causes a conformational change in the enzyme that changes the enzyme's ability to carry out its function with its active site. So a lot of times you can allosterically turn on or turn off an enzyme. That's what you're seeing in this diagram here. Oh, sorry, this is an inhibitor. You're actually seeing it turn off. So right now you have an active site that's working, and this substrate would be able to fit into it, right? The curved side of the substrate would be able to physically fit into the structure of the enzyme. But you allosterically add an inhibitor. The inhibitor binds to the enzyme and changes the shape of the active site such that the substrate can no longer fit in, right? You alter the active site and change the function. Not every allosteric process is inhibitory. There's some that activate enzymes. There's some that deactivate enzymes. There's a wild range of effects. Some kind of deactivate things. There's a lot of options, and those are very context dependent. So if you need to know about some specific context, that will be provided to you. So it'll be like something like, okay, there's enzyme A, which has an allosteric site for an inhibitor. Based on that, you say, okay, an inhibitor can bind and turn this off. If I say there's an enzyme that has an allosteric site for an activator, you say, okay, the activator will bind and turn that on, right? So that'll tell you a little bit of information about what is the base state of the enzyme without that allosteric ligand binding, and what is the effect of that allosteric ligand. So again, the key point here is that you're taking advantage of the idea that structure equals function and changing the structure of the enzyme and thereby its ability to carry out its function. Does that make sense to everyone? Okay, any questions? Lovely. All right, so that's allostery. Next, we're going to talk about post-translational modifications, or PTMs. In this class, we're really scratching the surface here. You can get in a lot of depth with post-translational modifications, but basically this is after the protein is made. So after the process of transcription and the process of translation, right? You've learned about, or we've talked about some post-transcriptional modifications like splicing, things like that. Those things happen at the mRNA level. This we're talking about at the protein level, so post-translation. So after the protein is made within a ribosome, you can alter the tertiary or quaternary structure of the protein by chemically modifying the amino acids that make up the protein, right? By adding functional groups. The big ones that we'll talk about are phosphorylation, ubiquitination, and acetylation, right? Phosphorylation is the addition of a phosphate group, which is negatively charged. Acetylation is the addition of an acetyl group, which has no charge. And ubiquitination is the addition of a ubiquitin, which is a small protein that also has no charge. So what you're doing there is by adding ubiquitin, you might be changing the size of something or the physical structure of an amino acid. By adding phosphorylation, you're making that amino acid negatively charged where it might not have been before. With acetylation, you're making something, the amino acid have no charge when it did have a charge before, right? So do you see what, so like through all these mechanisms, you're changing the structure of the amino acid and the chemistry of the amino acid, and therefore you're changing its ability to bind to ligands, or whether that ligands the reactants, products, substrates, whatever it is, whether we're talking about an enzyme or not, you carry out post-translational modifications to change the chemistry and structure of the amino acids and therefore change its binding ability. Does that make sense to everyone? Okay. One thing I do want to say is that a lot of people get in the habit of thinking of phosphorylation as turning something on, right? An enzyme gets phosphorylated and now it's in the on position. That's not always true. Oftentimes, yes, you will see that a kinase, which is an enzyme that phosphorylates another protein. So kinase will phosphorylate its target protein, and then that target protein, if that's an enzyme or whatever it is now in its quote unquote on scenario. But that's not always true. Sometimes phosphorylation is used to deactivate proteins or enzymes. So keep that in mind. Don't get in the habit of saying, okay, phosphorylation is always activating. Sometimes it's activating, sometimes it's not. Again, it's very context dependent. And when you need to use, like, think about this stuff in post-translational modifications, the context will be explained to you or given to you. All right. The other thing is these are reversible, right? You can remove these modifications after you've done them. If you add a phosphate group or ubiquitin or acetyl group, you can use another enzyme to take them off. Just as there are kinases to put phosphate groups on proteins or structures, you have phosphatases to remove phosphate groups from those same structures, right? Does that make sense to everyone? Any questions? Okay. Now, the last thing we're going to talk about today is nucleotide binding and nucleotide hydrolysis. It's kind of similarly to allostery, right? You can have nucleotides, main ones being GTP and ATP, that bind to proteins. Proteins that bind GTP are called GTPases. ATPases are proteins that bind ATP. And these proteins will have, like, GTPases will be able to cleave the third phosphate group on a GTP to make it GDP. So it'll go from GTP bound, it'll cleave the phosphate group off of the GTP, and then it'll remain GDP bound and let that phosphate group go. That can change things from being in the on position to the off position, quote unquote. So it can make it so that from an enzyme that's bound to GTP might be able to carry out whatever function it has, but then when it cleaves that phosphate group and goes back to being GDP bound, then it can no longer carry out its activity. The GDP or ADP, depending on the protein, we'll talk a lot more about GDP and GTP based switches and how they regulate proteins moving forward. But the GDP can then be like kind of kicked out, similar to the phosphate group, and a new GTP can come in, and you have this like little cycle that allows you to turn things on or off, depending if they're bound to GTP or GDP or ATP or ADP. Right. So this is essentially a special case of allostery, where instead of some inhibitor or activator, it's GTP or ATP or GDP or ADP. And you can go from GTP to GDP by just cutting off that phosphate. And you can go from GDP back to GTP by just getting rid of the GDP entirely and getting a brand new GTP. Does that make sense? There's a lot of G's. Any questions or concerns? All right. That's really all we have. Yeah, so thank you for coming. We will send out this recording somehow. And we'll also try and send out the slides as well. We're gonna stop recording now, but if you guys